All right, so the cell cycle, we're talking about what goes on in the life of a cell. So that's what the cell cycle means. And here we have this pie chart here that goes through the different parts of the cell cycle. If we start here with interphase, so from here all the way around is interphase. And during that time in the cell's life, it's just going through normal everyday activities. For instance, if it's a liver cell, it's going to be acting as a liver cell. If it's a skin cell, it's going to be acting as a skin cell. Okay, so whatever the function of that cell type is, it's going through its job. It's also going to be growing. So the G1 it actually stands for gap, but you can think of it as growth. Okay, so G1 phase is cell growth. Then we have the S phase, and S is for synthesis. Do you remember when we talked about synthesis reactions? And we talked about those are reactions that are building up something larger. So you start with pieces and it's building. So during synthesis, it replicates or constructs the genetic material, the DNA. It replicates. Then we have G2. G2, again, think of it as a growth. G stands for gap again. But there's more growth that occurs, and then the cell begins to prepare to divide. Okay? Now, there are some cells that cannot divide. Okay, red blood cells, for instance. Once they're produced, they cannot divide because they are missing something. And Dakota, do you remember what red blood cells don't have?
Right. That's what starts me in my mind.
They don't have a nucleus. Yeah, so when you look at them under the microscope, they don't have a nucleus. All right, so interphase is most of the life of the cell. And then we have mitosis. So mitosis literally means division of the nucleus. Okay, so that's when the nucleus is going to divide up its genetic material and produce more genetic material and put it in a location that it can be divided up, and then the cell will have material needed for the new cell. So then we have prophase, metaphase, anaphase, and telophase, which are the phases of mitosis. Okay. All right, so then we go through what happens in each one of these phases. Also, notice along the way that we have checkpoints. So here is a restriction checkpoint. And what this does is it looks at what's happening with the cell. It checks through things and makes sure that the cell, it has accurate information in it, that there's nothing wrong with the cell, so that it can go on and replicate the DNA, but it only wants to replicate DNA that is the correct DNA, right? If there's something wrong with it, then the cell would need to get rid of that DNA. All right, so this restriction checkpoint is where it's checking the DNA. Then this term here, apoptosis. Apoptosis occurs if there's something wrong with the cell and it can't fix the DNA, then it will kill the cell. Okay, that's programmed cell death, apoptosis. If the cell is okay to proceed, it can either remain as that type of cell, or it can go on and go through cell division. Okay, so if it goes on, then it's going to come around here to mitosis and go through those phases. So over here we have the stages of the cell cycle. We have interphase. Notice that interphase is not part of mitosis. Okay, interphase is before mitosis. Then we have mitosis. Mitosis is division of the nucleus. And then we have cytokinesis. Cytokinesis is division of the cytoplasm. Right, so we talked about interphase, what's happening there, and this replication of the genetic material is very important. When we get on to the next chapter, we're going to talk about how DNA replicates itself and how important that process is. So that happens during interphase. Okay, then we have the phases here with synthesis, G1 and G2. And remember, it actually stands for gap, but you can think of it as growth because there is stuff happening there. Okay, mitotic cell division. So here's where we get to mitosis and the phases of mitosis. Prophase. It starts out with prophase, and in prophase, the chromatin condenses. So chromatin is the DNA plus proteins in the nucleus. So chromatin condenses and forms chromosomes, right? Chromosomes it's just DNA and protein, and it forms a particular shape, which is like a long shape, kind of like a long string, okay? And if it's a replicated chromosome, it has two sides to it, okay? So chromatin condenses to form chromosomes. The centrioles, if you remember from the parts of the cell, the centrioles are the cylindrical objects in the cell, organelles, that are made up of protein, and those centrioles are going to move to opposite sides of the cell. The centrioles are going to start to form the spindle apparatus, okay? And what the spindle apparatus does is it attaches like little thread-like protein structures to each of the chromosomes, and it moves the chromosome around. Remember when we talked about how do animals that change colors, how do they change colors? I told you guys about that, and they move the little packets like an octopus or a chameleon, right? It moves these pigment packets around in the cell, and that's what allows it to be able to change colors. Okay, the same thing happens with the chromosomes. The cell can move the chromosomes around using these protein fibers. And it moves it so that they're going to eventually line up in a straight line. So that's going to be the second phase here, is metaphase, where the spindle fibers, which were attached from the centrioles, attached to the chromosome and a line in the middle. That's called the metaphase plate. Okay, so the spindle fibers move the chromosomes to the metaphase plate, and it's a straight line in the middle of the cell. Right? Then we have anaphase. And anaphase is where the chromosome, which has two sides to it because the DNA has been replicated, is going to be pulled apart. So the chromosome, one side of the chromosome is going to go to one side of the cell, and the other side is going to be pulled to the other side of the cell. And that's allowing the cell to be able to take that replicated DNA and section it off in two sections so that the two new cells can be identical to one another. Right? So those chromosomes separate and they're going to move to opposite sides. And then in telophase, things start to go back to normal. Then you start to go back to having two separate cells. Okay? So the chromosomes are going to decondense. They're going to go back to chromatin. The nuclear envelope is going to reform around the chromosomes. And the cell is going to continue to pull apart. Okay, so in cytokinesis, we talked about division of the cytoplasm. That actually starts here in late anaphase and continues on through telophase. So here we have those major events of mitosis. This is a great table for you to study. I think there's one of your learn questions also where you need to go through and list the events of the cell cycle. Also in your lab book, you had a section there where you needed to list the events of the cell cycle. So this is a great table to help you with that. All right, so here's slides of cells going through the cell cycle, right? It shows those details that we talked about here with what's happening with the nucleus, the chromosomes. Here we can see those spindle fibers attached to each side of the chromosome, lining them up in a straight line. And then here's that metaphase plate, that straight line there, where each side of the chromosome is attached to a fiber. And then here in anaphase, where you see that the chromosome has been pulled apart, and this side is being pulled over here, and that side is being pulled over there. Okay? So what you're going to end up with in mitosis is two identical daughter cells. So it starts out with one cell, and the DNA is already replicated, okay? And remember that happens in S of interphase, the DNA is replicated. And then when it goes into mitosis, that replicated DNA, which is, so right here on the chromosome, this side is identical to that side, okay? And then it gets pulled apart so that this side is going to have the same identical information as this side. And then here we see cytokinesis occurring with the cleavage furrow, which is going to pull the, it's like proteins in there that squeeze apart the cell, and then you end up with two separate cells, which are then going to go back to interphase. So it cycles back around. All right, so cytokinesis, division of the cytoplasm. As I said, it starts in anaphase and goes through telophase. And the contractile ring of actin filaments, which is squeezing the cell apart, that's proteins in there that squeeze the cell. All right, so the original cell here is going to be identical to the two daughter cells. Okay, you don't end up with three cells, right? You end up with one cell that has divided in half and produces the two identical cells. Okay, there is control in cell division, and that's important because when cells lose control, that's when they produce cancer. Okay, so the control there allows the cell to be able to divide when needed, but it doesn't just keep dividing and dividing and dividing, right? So that's important. Frequency of cell division is regulated. Skin cells, for instance, are going to continuously throughout your life reproduce, right? If you just scratch your skin like that, you see those top cells coming off, right? Or if you have dry skin, that's those dead skin cells. They're constantly replaced and push up so that the layers are continually pushing up. Neurons, however, they divide a specific number of times and then they stop. They don't continue to divide throughout your life. So your brain cells, right, once your brain is developed, they're not going to continue to divide. That's why brain damage after a person is fully formed is such a critical thing, because it's not going to just be able to replace those cells, right? So you got to take care of your brain, wear a helmet, things like that you can do to help to take care of your brain and protect it. So the chromosome tips, which are telomeres, okay, that they shorten with each division of the cell. So each time the cell replicates the DNA, that little segment of the chromosome gets shorter. And when it gets to a certain length, it's going to stop replicating that DNA. It's not going to be able to continue to divide. So that's the telomere. That's what causes aging. The telomere theory of aging is that those telomeres, the little segment on the tip of the chromosome, when they get to a certain length, shortness, they no longer divide. And so those cells are just going to have to age after that. They're not going to be able to reproduce themselves. Okay? Different levels of proteins in the cell are going to control the cell cycle. So there's different enzymes in there that are going to be acting. Cells divide to provide more favorable surface area to volume ratio or relationship. So that's important in that the cell has to be able to get all of the needed materials into the cell through the cell membrane. So if a cell gets really big, then the surface area is not enough to be able to bring in the necessary materials to keep that cell alive. So by dividing the cells up into smaller components, it increases the surface area, okay? And if we think about that, like here's a box, right? So this box has this certain surface area to it, right? But if you divide that box up into smaller boxes, then each one of the smaller boxes has its own surface area on the interior, right? So it's not just the exterior of the box. There's a lot of little tiny boxes in there. So that increases the surface area, okay, the surface area to volume ratio. So that's important for cells to be able to get those materials in. Then hormones and growth factors. So as you're growing, right, you know that as you're growing and developing, your hormones are going to different areas of your body and stimulating you to grow. Okay, growth hormone comes from your pituitary gland, and it goes to your bones and causes them to lengthen, right, and goes to different areas of your body, causing you to grow, right? So those are external controls that are going to go to the cell and stimulate them to have that reaction. Then we also have this thing called contact inhibition, okay? So when cells get into contact with other cells to the point where they're getting crowded, basically, they're going to stop dividing because cells don't need to be crowded and pushed together, okay? So they need to have, it's like being squeezed, right? They do better if there's other cells around them, but not squeezing them, okay? So that contact inhibition, the healthy cells stop dividing when they become crowded. Tumors result when there is a loss of control over cell division, okay? And there's different factors that can cause that loss of control. Right? So there's two types of tumors. Okay? There's benign and malignant. Okay? A benign tumor, or if you had a biopsy done and it came back as benign, right? That means that it is not dangerous at that point, that it is encapsulated in a local area, and it may just be kind of like a mole. Let's say you have a mole removed and they biopsy it, right? And it comes back benign. It's just a mole. There's nothing dangerous about it. But if it comes back as malignant, that means it is invasive. And benign tumors can turn into malignant tumors. Okay, so the malignant tumor is invasive. It's cancerous. It can spread and metastasize. Okay, metastasize means that it can break off from that original area and move to another area of the body and start a new tumor. This happens especially with breast cancer. Okay, breast cancer starts off in the breast where there is one cell that has DNA in it that's damaged, and that one cell begins to divide uncontrollably, right? So it loses contact inhibition, and it starts to divide and divide and divide, and then it metastasizes. So one cell breaks off from that tumor and travels through the lymph and through the cardiovascular system to another area of the body and lodges there and then begins to grow. Many times it can go to the brain and start a brain tumor. So it goes there, it begins to start a new tumor in that location. Also, the lymph nodes are another common location for metastatic tumors, where it started somewhere else and then cells break off and go to the lymph nodes, go to the brain, can go to any other organ, because they're all connected, right, by the cardiovascular system, and start a new tumor in that area. Okay, so malignant tumors metastasize. And also that they're invasive, they start to grow down. So they're not only like if you had a mole on the surface of your skin that's just growing up, right? And at that point it's still benign, but the malignant tumors grow deeper into your body, and they have like little roots that grow out from it. Okay, two major types of genes that cause cancer. We have oncogenes and tumor suppressor genes. Okay, so oncogenes are, we all have these genes, right? They're called proto-oncogenes when they're just normal genes. But if that genetic material gets altered in some way, then it becomes an oncogene. Okay, and so we know that there is a genetic basis for cancer, right? We probably all have heard of people that have smoked for years and years and they never got cancer. They, died of something else, or they got in a tanning bed their whole life and they never got skin cancer. They died of something else. So there's, all these different things, genetic factors that play a part in that. Because if you carry a certain gene, then it's more likely to mutate to become an oncogene. Okay, so there's certain factors in there in the DNA is more easily altered and changed into an oncogene. Okay, so those oncogenes are abnormal form of the genes that control the cell cycle that we've talked about before, that are going to be checking on that DNA, making sure that everything's okay, making sure that the cell needs to continue on and divide. Okay, the tumor suppressor genes, same thing. They're normally they're going to be limiting mitosis, but if they are inactivated or removed, if there is some damage to that particular section of the DNA, because that's what a gene is, it's a particular section of the DNA that codes for a particular protein. Okay, so if something happens there to that tumor suppressor gene, then it is no longer suppressing the tumor, then that area is going to continue to divide and grow uncontrollably. So here's a photograph here of the green cells here represent the cancer cells, and then these here are the normal cells, right? So what's the problem with having too many cells, right? You wouldn't think that that would be so dangerous and so lethal. But the problem with having these tumors and having too many cells in that tightly compacted area is that the tumor cells are not acting as normal cells of that tissue. The tumor cells lose their function, and all they want to do is divide, 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 okay, produce more and more. So the tumor cells are not working. Let's say this was in your liver or this was in your pancreas, right? They're not working as liver cells. They're not working as pancreatic cells. They're only dividing and metastasizing, and that's all they're doing. Okay? Now, it becomes very lethal when the function of the organ is no longer, the organ's not going to be able to function any longer because it's taken over by the tumor. Okay? Also, it becomes lethal when the cells have metastasized to an area that is very critical, like lungs, like the brain, right? Where your body's not going to be able to function if it has these tumors in there that are no longer have the function of that organ. Okay, so here's those steps in the development of cancer. So we start out with healthy, specialized cells, right? And something triggers a change in the DNA. What are some things that you know that can cause cancer? Things that you've heard of that can cause cancer? The sun, right. Exposing your skin to the sun and getting sunburned damages your DNA, okay? So that damaged DNA is then going to go through that cell cycle, and it can get fixed right at the checkpoint. The checkpoint can say, hey, there's something wrong with this DNA, let's fix it. So it can fix it, or it can get rid of the cell, kill the cell. So when your skin peels off after sunburn, right, that's what's happening. Those cells are damaged and they're just going to die, okay? But cancer can develop if those cells make it through the checkpoint without being fixed or without, dying. So something triggers that. We've got things like tobacco smoke, excessive eating of certain chemicals like in preservatives, okay? There's pesticides that cause cancer. There's things in the air, environmental things. There's cigarette smoke. There's alcohol. So many different things that are carcinogens, right? They can damage or mutate the DNA, right? But our bodies are really good at fixing that damage most of the time, okay? So most of the time, we're going to be able to fix that. Sometimes it's just a spontaneous mutation, like with people that carry the gene for breast cancer, right? It just happens. That mutated gene is there, and everything goes along fine for many years, and then all of a sudden breast cancer develops, right? It wasn't because they were actually exposed to cigarette smoke or anything like that. It was just that the cell cycle lost control because of damage to the DNA. All right, so loss of cell division control, loss of specialization. The cells can't work as the cells need to anymore. Mutations occur, right? So it just continues to, as it's dividing uncontrollably, to mutate. Here we have angiogenesis occurring. So angiogenesis is when blood vessels grow to a tumor. So that's another problem with cancer, where it's taking away the blood supply to the healthy tissues, and it's carrying that blood to the tumors. And the tumors allow to grow and grow and grow. So that's called angiogenesis. And here you can see this capillary network, and that can cause problems with trying to remove a tumor also, because it's so easy if, let's say, they're trying to cut out this tumor here, and a cell breaks off as they are trying to remove the tumor. That cell is then going to go through the cardiovascular system, through the arteries and veins, and it's going to go somewhere else and set up a new tumor. And so then it's just spread. So surgery, in some cases, they can do surgery, and it's very, successful. sometimes they have to damage the tumor with radiation or chemotherapy. And the problem with those therapies is that it also damages healthy tissue. And so it makes the person very weak, a lot of times immunocompromised, things like that. So there's a lot of different factors that they look at in determining the course of treatment for cancer, for tumors. Any questions before we go on? Okay. All right, so the next thing is stem and progenitor cells. So we have the term differentiation. Okay, so that is specialization of the cell. So once a cell is produced, then it specializes. It's going to become a certain type of cell. So we think about when you have a fertilized egg, the zygote. Okay, the zygote is an undifferentiated cell. It's just a fertilized cell. Okay, then it begins to divide, and it divides and divides. So from that one cell, all of the cells, all of the tissues and organs of your body are produced. So along the way, there has to be specialization that occurs to produce liver cells, to produce brain cells, to produce pancreas cells, to produce muscle cells, right? So that specialization is what we call differentiation. So stem cells have the ability to divide to form new stem cells, okay? And stem cells are undifferentiated. They have that self-renewal. A stem cell can also divide to produce a stem cell and a progenitor cell. Okay, and a progenitor cell is a cell that's just slightly more determinate. Okay, meaning that it has a certain pathway that it's going to go down. Okay, it's not just going to keep dividing and producing stem cells. It's going to divide and produce a particular type of cell. Right? So the progenitor cells are partially specialized stem cells. They're the daughter of the stem cell, and they can divide to produce a restricted number of cells. They're committed. They're more determinate. Okay? Then we have these totipotent, and these are cells that can specialize to become any cell type. Okay, for instance, the fertilized egg, right? As it divides, it's going to produce all of the cell types, right? Pluripotent, daughter cells that can become a limited number of cell types. So once the stem cells divide and produce the progenitor cells, then they're going to become more and more determinate as to what they can produce. Okay, so here we have an example here with a stem cell. And this is hematopoietic stem cell, which is going to be producing blood. Okay, and here we have the self-renewal, so it's going to make another stem cell, right, so that property is retained, but then it's also going to produce a progenitor cell, and the progenitor cell is then going to divide to produce these different white blood cells that are, so with each division here, it becomes more and more specialized. Okay, differentiation, maturation, and specialization of cells. So again, if we go back here to the fertilized egg is called the zygote, okay? And the zygote is then going to begin to divide and produce these stem cells. Then the stem cells are going to divide and produce more stem cells and also progenitor cells. Then the progenitor cells are more determinate, right? So they're going to produce more progenitor cells, but these progenitor cells are going to have a certain pathway that they can go down. So for instance, here we have skin cells. Here we have progenitor cells that produce neurons, right? So it becomes more and more specialized. Okay, we hear about stem cell research, right? And stem cell research is research where they're trying to use these stem cells to help to repair tissue that can't repair itself, right? So, for instance, with the nervous system, right? So we know that the nervous system, the brain cells, once they're produced and they mature, they can't continue to divide. So if you have damage to the nervous system, it can be detrimental. It can be, life altering. So the stem cell research, they're trying to figure out ways to use stem cells, and there has been some successes with it, to be able to use stem cells from, say, a donor, or from now people have begun to save umbilical cords. Like, you can have your umbilical cord saved in a special bank thing, and then if something happened to your child later on, they developed some disease or had an injury, then they could take some of the stem cells from the umbilical cord, and they could try to use that if the technology is available later on to be able to use it. They don't have what's really needed at this point, but people are kind of banking on that for later on. It's very expensive though, and we don't actually have the technology for it at this point. But things are, being produced every day, so research goes into that. So the donor stem cells, umbilical cord cells, stem cells from the patient, so either from a natural site such as bone marrow, because you have cells in there that are continually dividing to produce blood cells. So those stem cells, or program differentiated cells grown in a cell culture. Okay, so that's another way that stem cells can be produced, is by stimulating their genetics to continue to divide and to produce certain products. Right, so we talked about this term apoptosis, right? So if the cell can no longer survive, or if it shouldn't survive, right? If the DNA is damaged, then it needs to be killed. So that's programmed cell death. It's a normal part of development. For instance, here, the webbing between fetal fingers and toes. So as a fetus, you have webbing between your fingers and your toes. And at a certain point in fetal development, that webbing goes away. And so the way that it goes away is that those cells in the webbing, they're programmed to die at a certain point. So that's apoptosis. Okay? It's protective. It peels away damaged skin cells after a sunburn, as we talked about. When the DNA is damaged, that's your protection, is to kill those cells so that they don't replicate. Okay, necrosis, cell death from damage. That's not a normal, living process that has something's damaged. And here you have that sunburn there with the cells dying. So the death receptor on the doomed cell binds to signal molecule, and it starts to cause the cell to form these little things called blebs. So it's like little bubbles on there, and it bubbles off, breaks apart into little cell fragments, and then you have your white blood cells that are going to engulf those and take them in, break them down, and recycle the little blebs there. Okay, cytoplasmic organelles, long description. Okay, so proteins are synthesized in the rough endoplasmic reticulum, right? The ribosomes that are attached to the rough ER are going to be synthesizing proteins, right? And the fats in the smooth ER, sugars in the smooth ER, and the Golgi apparatus. And those products are going to be sent to the Golgi apparatus for packaging and modification. The sugars and proteins are released from the Golgi apparatus in vesicles. Vesicles are like little bubbles in the cell. And the vesicles are then going to be secreted from the cell. They're going to go to the cell membrane and open up and be released to the outside of the cell. The fats are going to travel directly from the smooth ER to the cell membrane, where they can become encased in a layer of the cell membrane and exported from the cell. Okay, so that's what's happening, the machinery inside of the cell as it is, those organelles are working in the healthy cells.